It's my big privilege to welcome Tim Hunt again in Hungary, and we are really very gifted that Tim Hunt came to Hungary again. And I just start again with the background, with the grandfather's part. So Tim Hunt's paternal grandfather was a doctor, and an old Victorian pharmacy, and actually his maternal grandfather was a timber merchant who improved wood from South America. And if we go further concerning his family part, which, as I mentioned in the earlier introductions, it's highly, highly important. His father was a lecturer in paleography. They studied the ancient and historical handwriting, and during World War II worked at Bush House in London, the headquarters of BBC World Service Radio. So concerning a schoolboy in Oxford, he was taught Latin at home by a governess and attended the infants department of the Oxford High School for Girls. Accepted into a dragon school. Now it's most probably called Harry Potter School, but at that time it was the Dragon School, as a private school where he developed his interest actually in biology, and later on at 14 he moved to Magdalen College School, Oxford. Of course, after high school he didn't stop. He went to universities at an early age. In 1961 he was accepted into Clark College, Cambridge, to study natural sciences, and later on he continued working, and in 1966 he worked in a laboratory at Albert Einstein College of Medicine. He loves animals. I learned in the last couple of visits he loves birds, but of course there are some experiments he realized during his career, and actually in school he and his classmates dissected his brother's pet rabbit, but luckily after he died, and which was a treat after all the formalin-fixed dogfish. So if you have pets, of course later on it's a possibility to go forward. And his PhD focused on hemoglobin synthesis in intact rabbit reticulocytes. Summers in Massachusetts, actually he regularly spent summers working at the Marine Biological Laboratory at Woods Hole, so he continues his interest in science. And there are the important animals, number 2 in his life, that it was at Woods Hole around 1982, the sea eggs, and this was the time when his model organism that he discovered the cyclin proteins, which play a crucial role in regulating the cell division cycle. Important animals, number 3. So he continues his interest in nature and science, and concerning the eggs of the frog Xenopus, he continued his research in the cell division, and he demonstrated that cyclins were present in the cells of most organisms as well. So in the 90s, the Cancer Research UK London Research Institute hired him, and he started really working there very efficiently. And in 1993, the book, The Cell Cycle, was introduced by him, and I think this is one of the best known, well-known books concerning the cell cycle at that time. So not surprisingly, because his major discovery in the cell divisions, he received the Nobel Prize in 2001, and moreover, he was just knighted in the 2006 birthday honors of his service to science. On the other hand, the National Portrait Gallery in London houses 2 photographic portraits of Sir Tim Hunt. And in the 2008 portrait, actually this is also very famous, holding a model of a CDK2 cyclin, which actually he is very well known. Concerning the awards and honors, of course, besides the Nobel Prize, he received many distinguished prizes during his life. But again, like I introduced with the earlier speakers, family is always one of the key points. So, and he married to the immunologist Mary Collins, and they worked a lot, and Mary passionately advocates for developing young researchers, like he's always visiting us. And I mentioned that we have a privilege to have him because this is the first time that he is in Hungary in this program. He was here in 2012 when 9 Nobel laureates arrived at the same time to Szeged when we established this school, and after in 2015, 17, and also now in 2025, he joined us. So he, of course, has wisdom for young scientists as well. So this is highly important, and he believes very much in giving power to the young and not putting them under. And he was given full autonomy and authority at an age of 27. And I think this is highly important for young generation to receive the possibilities to act. Finally, it's a great privilege and honor, Tim, to welcome you again in Hungary. And thank you for continuously visiting us. And I hope that this is also a continuation, and we will be able to welcome you, not only this year, but in the forthcoming years as well. Thank you so much for coming.
That's very nice. Thank you, Peter, for inviting me back. I love coming here. And there's always something interesting and new. Today, or rather yesterday when we first arrived, we saw some amazing birds from amazing high-tech, you know, this was just extraordinary. It was a Zen-like experience of watching these giant birds at close quarters. They couldn't see us because we were behind a special place, you know. It was just wonderful. Then I saw the most amazing dissection of a blackbird by a prize-winning taxidermist. And I mean, this was just extraordinary. He used exactly the same kind of scalpel as I had used for dissecting my brother's pet rabbit. It was terrific. Anyway, and also, you know, the fish soup and the goulash are pretty cool, too. My wife is very disrespectful, actually. She says, she thinks of me as the People's Laureate because she says, if I can win a Nobel Prize, anybody can. and I think that's absolutely true. You don't, let me assure you, you have to be a genius. You just have to sort of follow your nose, okay? And I'm going to try and tell you about that nose-following business with the story of my life. So, as Peter mentioned, I grew up in Oxford. Now, an interesting difference between Oxford and Cambridge, and I went on to Cambridge, is that 2/3 of the people in Cambridge are scientists, whereas 2/3 of the people in Oxford are not. Okay, so Cambridge is the place to go and do science. There's absolutely no question about the tradition of scientific discovery, from the discovery of the atom and the discovery of the structure of DNA. Not only how the two chains were put together, but also the chemical structure of DNA was determined in Cambridge. Alexander Todd and, oh gosh, what was his first name? I knew him. Brown was his last name. They were very cross because they always felt that Watson and Crick got too much credit, that working out the chemical structure was also a pretty fine achievement, and of course absolutely necessary for Watson and Crick. Anyway, so why did I go to Cambridge? Because, partly to get away from home, and partly because it was the place to do science. And I passed the exams well enough. I mean, I'd sort of like to tell you some stories about my time there, but there isn't really time. And then went into a lab, and the reason I chose the lab I chose was very simple. It was run by the guy who'd been my undergraduate biochemistry tutor. Why did I choose him? Well, one time I had a terrible cold, and I went to a tutorial. We call them supervisions in Cambridge. And he said, "Oh, Tim, you look terrible. Here, take this." And he handed me a glass of whiskey. So I knew he was my kind of guy, you know? And indeed, and he was a terrific mentor. So I joined his lab, and he had a large lab, and he was the only person in the biochemistry department who worked on what we would now call molecular biology. But it was very disparate. I mean, people worked on DNA synthesis, RNA synthesis, and Asher himself worked on protein synthesis. Now, at that time, Jacob and Monod had just proposed their model for how gene regulation was controlled, how transcription was controlled. Messenger RNA had been discovered while I was an undergraduate, and it was perfectly clear that although the real action was in the control of transcription, I sensed that that was far too crowded a field, and my chances of making any significant contribution were as close to zero as made no difference. So I decided to work on translational control because that seemed, and I started out trying to do something which failed miserably. I won't explain what it was. And after about 6 months, I had absolutely nothing. And then luckily, I went to this conference, which was just down the road in the chemistry department, and I heard these two talks. First talk was by Henry Borsuk, and what he talked about were two things. One was the control of hemoglobin synthesis, and he and his colleague Jacques Roux had discovered that if you removed iron from the cells that were making hemoglobin, reticulocytes they're called, the immature young red blood cells, not only did they stop making the heme, because if there's no iron, of course they can't complete the synthesis of heme, they also stopped making the protein. And that was very surprising. So there was coordination between the heme and globin synthesis. And in an attempt to, how do I go backwards? To understand this, the other talk that made an impression, well, I ought to mention this other thing. You notice his title, Early Development of the Echinoid Egg. What's echinoid? What is an echinoid egg? Do people know what an echinoid egg is? It's a sea urchin egg. And he had discovered that when sea urchin eggs are fertilized, they increase the rate of protein synthesis. Now, there are no sea urchins in Cambridge, so there's no possibility of studying that, but it planted a seed in me. Now, as far as the heme regulation of globin synthesis, Vernon Ingram had been working on this and had come up with a very interesting idea. He showed data which he claimed indicated that what happened was, if you imagine the start of the globin messenger RNA is here, and that I'm a ribosome making hemoglobin, well, one of the chains of globin, right? I start, and the globin chain comes out of my back like this, getting longer and longer, and finally you get to the place where the heme should come in. And what Vernon said was that if the heme wasn't there, the ribosomes just stopped and waited for the heme. I mean, how the hell did the ribosome know whether the heme had arrived or not? I mean, it was pretty incredible. And then if the heme arrived, then it could go on and finish, okay? Very interesting. I went back to the lab and said, gosh, look at this. And somebody who was, it was probably Lou Reichart, who was much cleverer than me, pointed out that actually he totally misinterpreted his data. Great scientist, the discoverer of the mutation in sickle cell anemia, and he'd simply got the interpretation of his own data wrong. He hadn't thought about it sufficiently detailed. So we thought, being brash young first-year graduate students, thought we could do it better. And we could. And indeed, when we finally wrote the paper that definitively proved him wrong, he was absolutely wonderful. I met him in a restaurant very close to where we live now. It's called Pescatori. It's closed down. And he was the editor of the manuscript. And he was so nice about it, encouraging. He was really, really pleased that somebody had taken him seriously and proved him wrong. And I think that was a sort of, there was a lesson in there somewhere. Now, Lou was trying to make a cell-free system, and it was very easy to make cell-free systems from these cells. So there they are. They're called reticulocytes because they have a sort of lacy network of blue dye in them if you use the right kind of dye. And amazingly, if you simply add distilled water to those cells, they burst open. And even more amazingly, if you then spin out the junk, the supernatant, which is basically 50% diluted with water cytoplasm, but everything else is there, it continues to make protein at the same rate as they would have done if you hadn't broken the cells open. How that's possible, considering it takes about 200 different components. I mean, you would think that you would slow down by factorial 200 or something, but it doesn't. It's very, I mean, I don't know, I still don't really understand that. Anyway, but anyway, Tony and I collaborated on, and we showed that actually the ribosomes didn't form Qs when you left out the heme, and we discovered one or two other things. And in the middle of that, I went to this meeting, the second meeting. So one of the lessons is that always go to meetings, and because you'll meet some interesting people randomly. And this guy, I'd never heard of him. I met him randomly. He's much older than me, and it turned out he was the chair of the Department of Medicine. But that didn't matter because, you know, we were both interested in the same things. And, you know, we were both struggling now with this question of how heme controlled globin synthesis. And he said, you know, Tim, why don't you come and join my lab when you finished your PhD? And I said, to hell with finishing my PhD. Couldn't I come and work in your lab this summer? And he said yes. And by the time I'd taken a slow train back from Yugoslavia to London, there was an airline ticket. In those days, the tickets were not bits of virtual PDFs. They were actually pieces of paper, funny things, waiting for me. So I went and spent 4 months in the summer. And I liked working in his lab, and I liked the people. So it was no problem deciding that's where I should go for the postdoc because, you know, he was one of the very few people in the world who was interested in this problem and had been for ages. And so it was good fun. And there is the sort of typical thing. This is a cell-free system now. Time running along the bottom, protein synthesis up the side. And if you can see, if you add heme, it works. And if you work out the rate, that's pretty much the same rate as they would have, you know, normal rate of protein synthesis. If you leave out the heme, however, it starts off perfectly fine, and then after about 5 to 10 minutes curls over and goes to a low rate, typically 10 to 20% of the initial rate. And if you add back the heme after 5 minutes, you know, it recovers. So this is a reversible control. So the question is, how did it work? What was going on? Well, basically, I kept on repeating that experiment. And if you repeat the experiment, it's very robust. It always worked. But it didn't lead to any insight in how it worked. And so I started getting very frustrated and doing other experiments after my technician had gone home in the evening and at weekends. And I thought I would map the genes in poliovirus because I could use the technique I'd used before. So I found there was somebody in the building who worked on poliovirus. She was called Ellie Ehrenfeld. And I got some poliovirus RNA from it, looked to see whether it got translated, and it didn't. Correct result, by the way. So why not? Well, it did get translated in human cells, so she had some human cells infected with poliovirus. That's how they grew the stuff. And so I asked for some poliovirus-infected cytoplasm from Ellie, added it in, and far from getting new synthesis of poliovirus, what actually happened was this extract inhibited protein synthesis exactly as though I'd left the heme out. And what was it? Well, it turned out it was big. It sedimented about 20S. It was really quite a large thing. And it resisted all enzymic treatments. So that was very puzzling. What the hell was it? It wasn't a protein. It wasn't DNA. It wasn't RNA. We began to fear that it was some horrible complex carbohydrate. And it wasn't present in control cells. Uninfected HeLa cytoplasm was totally innocuous. So that was really, really interesting. It took us about 6 weeks before we realized that actually the double-stranded RNA is resistant to ribonuclease. And then when we realized that, we went to Jerry Hurwitz, who was a nucleic acid chemist who trained with Arthur Kornberg, and said, Jerry, what can we digest double-stranded RNA with? He said, it's a micrococcal nuclease, and don't forget to add the calcium. So we added micrococcal nuclease, and that indeed did away with it. So then we knew what it was. Then I got some double-stranded RNA, and I ordered some double-stranded RNA from the Sigma Chemical Company, added it in, and did a dose response. And that was a funny result, because according to the dose response, I'd put the tubes in the scintillation counter the wrong way, because the highest dose of double-stranded RNA did not inhibit. If you add a lot of double-stranded RNA, it doesn't inhibit. But if you add a little bit of double-stranded RNA, it does inhibit. Well, haha. So then I repeated it, and that was the right result. So the dose response curve for double RNA, this is inhibition, this is normal synthesis. It went. So what does that mean? Well, I mean, the answer, if you think about it, is obvious. It meant that two things had to bind to the double-stranded RNA and talk to one another. And if you added too much double-stranded RNA, one thing would bind to that double-stranded RNA molecule, that other thing would bind to that other double-stranded RNA molecule, and they simply couldn't talk together. They have to sit close together and talk to each other. That didn't emerge until years and years later, but I already knew the answer. And we had a very funny fight with a guy called Ray Kempfer, who claimed that the way it worked was sort of mimicking the start of the messenger RNA. But actually, we knew he was wrong because the inhibition was catalytic. One molecule of poliovirus replicative form double-stranded RNA could inhibit synthesis by, I don't know, 10 million ribosomes or something. One molecule of double-stranded RNA was enough to completely kill a HeLa cell, basically, by this mechanism. And then I got involved in interferonology and stuff like that. It was really very, very interesting. And, but it wasn't going to get me a job. And it wasn't the main thing. And we didn't solve the heme problem. So I went back to Cambridge, where I had a fellowship. Interestingly, I took a 5-fold reduction in salary. In Einstein, I was earning $11,500 a year, which was pretty decent salary in those days for a postdoc. Now they get lots more. But I went back to earning, I think it was £850 a year in Cambridge, which was barely enough to live on. But fortunately, I had a college fellowship, so I got free dinners, and that was a help. And more to the point, I sort of rejoined my former colleagues in the Corner lab. Corner by this time had left. And Jim Watson says you should always try to work with people who are cleverer than you are. And I followed that advice and joined forces with Richard Jackson. And we had a wonderful collaboration. Lassie's no longer with us, and I miss him terribly. And by this time, I discovered that the way the inhibition worked, there was initiation was inhibited. And Richard and Tony had worked out that initiation of protein synthesis required a special initiator tRNA that used methionine. And so we switched to labeling the cells with methionine. And I'm going to show you some real live data here. And so these dotted lines are the methionine label. Small ribosomal subunit, the large ribosomal subunit, single ribosomes, and radioactivity in the dotted line. You see here, these are here's the uninterrupted state. The big peak over the 40S ribosome peak, which people had sort of noticed and said, oh, well, 40S ribosomes are sticky. But we realized, because we did the experiment, that when you left out the heme, this peak disappeared, and the peak disappeared before protein synthesis stopped. So that strongly suggested that whatever that peak was, it was a precursor. And indeed, it turned out that the way that protein synthesis was initiated was that you loaded the small subunit with initiator tRNA, and then that went looking for the start of the message. And when it found the start of the message, it stopped and waited, and then the large subunit came on, and then protein synthesis could start up. So now we had something to go on, and actually that was really the key to progress. And then the lab burned down. Now, you might think this would be a bit of a setback, but actually it was the best thing that could ever have happened to us because we had to move to a new lab, and it only took 6 weeks. The last thing to come, and it was a sort of rate-limiting component, the pH meter from, what were they called, radiometer, was the last thing to arrive. And without a pH meter, you can't make up buffers, so we were sort of stymied for 6 weeks. And we moved to opposite the great molecular biology lab. It was a sort of teaching lab in the hospital, which was 100 yards away from the LMB, which has 20 Nobel laureates in it. And Max Perutz was terribly kind to us and said we could use his stores. This was fantastic because the stores were really, really good. And the way it worked, there was a very complicated administrative system. You go in and say, can I have 10 grams of ATP, please? And there was a book with biochemistry written on the cover, and you wrote down 10 grams ATP. That was it. And you went away with the bottle in your hand, and everybody else, I mean, this was administrative heaven. It was awful. when I got to ICRF, you know, I mean, the number of forms you had to fill in to order something was just astronomical and stupid. And Max really made it easy to do. He was lowering the activation energy to doing experiments. Very, very important in my view. Anyway, and perhaps more importantly, he said we could have tea in his canteen. And the canteen, of course, was full of amazing role models. I mean, you know, so I knew all these people quite well because they would come and sit at your table and explain what they were doing, and they took you seriously as a young scientist. And Francis Crick in particular, who was our hero, I guess, because he sort of seemed to know everything, he was very concerned, to make sure that everybody was on board when he was explaining what he was doing. And he had a wonderful, simple way of explaining things. And the other thing that I learned from this, and you know, I mean, the other thing is that there are all these youngsters. Now, we didn't think we were in the same league as Sanger and Perutz and that lot, who were sort of, you know, godlike heroes on a pedestal. But actually, it turned out to be sort of quite, you know, we had interesting problems. We would explain to each other where we were, what we were thinking. We said, you know, and we criticized and helped, and I think we're all working on different things, so it wasn't at all competitive. It was just that, you know, people were working on ambitious, interesting things and making progress. And I used to organize a seminar where we'd find out who had a good story. And one of the rules of those seminars was that A, you had no slides, just had a blackboard, and B, anybody could interrupt at any point, so that it was always absolutely clear that people understood really properly understood what people were talking about. Super important, I think. Anyway, so this was very good, and partly a result of sort of having all new equipment and all new things and wiping out all the data, which was pretty foggy, and we figured out what it was. So anyway, we discovered that it was a protein kinase, which phosphorylated the initiation factor that bound the methionine. So everything just went click into place, and a huge amount of weird phenomenology was instantly explained. Now, what we didn't realize, or what we hadn't really fully appreciated, was actually there's not just one protein kinase. We now know, years later, there are actually 4 of them. And I think I only discovered this about a couple of years ago, trying to explain it to an audience rather like this. But actually, I think we made a big mistake in how it worked. I mean, the idea was that the heme kinase was a protein kinase that was inhibited by heme. No heme, the kinase turned on spontaneously. I don't think that's right, and I'll explain why. So a few years after we'd done this, 2 or 3 years later, Severo Ochoa, the great Spanish Nobel Prize winning biologist, published this paper which said that the way it works was by this sort of cyclic AMP cascade. Now, we knew that couldn't possibly be right, because actually high levels of cyclic AMP stimulated protein synthesis, it didn't inhibit. So it couldn't possibly be right. And I went to work with a guy called Phil Cohen in Dundee, got some purified cyclic AMP-dependent protein kinase catalytic subunit, added it in, had absolutely no effect. So Ochoa was clearly wrong. But how can he be so wrong? So we read his paper and we realized that he'd bought the enzyme from Sigma. So we bought the same prep from Sigma, and sure enough, the stuff, it said cyclic AMP-dependent kinase on the label, and sure enough, it inhibited. But it wasn't the kinase. We knew that. It was something else. What was it? The answer was, it was the bovine serum albumin carrier that they added to these preparations. So we tested bovine serum albumin, and that didn't work. So that was pretty peculiar. And then we further delving revealed that the Sigma Chemical Company first boiled their BSA to kill proteases. And when we boiled the BSA, it inhibited. So by this means, we failed to discover the unfolded protein response, which is what we had actually discovered, because we couldn't conceive of it. And we never boiled any other proteins. And it wasn't really what I was working on at the time. This was an undergraduate project. But now, years later, it seems to me that what is actually happening is that when you make globin in the absence of heme, it doesn't fold up correctly. You get a sort of wishy-washy, it's probably unfolded actually, don't really know. And that's what's inhibiting. And I suggested this to the woman, Jane Chen, who worked on this heme-regulated inhibitor, and she didn't like the idea at all. But then just recently I discovered in literature, I'm pretty sure I'm right, because it's been discovered that that's what's going on. Well, I can't do any experiments anymore, so I don't know. So anyway, that's sort of, yeah. So you win some, you lose some, is the moral of this story, and it doesn't really matter. So going back in time, we discovered this thing, we'd worked it out, we felt very pleased, but the problem was what to work on next. Because I think one of the big problems, if you're a successful scientist, is you have a problem and you solve the problem, you don't have a problem anymore, right? And finding a good problem is much easier said than done. Because a good problem is something that you would know when you solved it. I mean, that heme regulation thing is a very, very good example of that. I mean, it was confusing. It took a long time, 10 years more to solve. But we sort of knew when we'd solved it. I mean, there's still lots of details to be worked out, but basically that was the answer. And, there are a lot of things. I've also worked on a lot of projects where you wouldn't have really known when to stop because it didn't really have an end point. Anyway, I remembered that talk I'd heard about sea urchin eggs from Borsuk and invited Tom Humphreys, who was the only person I could find in the literature who still worked on it. And by pure luck, it turned out he was a very keen cyclist and wanted to go on a cycle ride when he arrived in Cambridge for the meeting. And he said, could he rent a bike? And I said, no, you have to borrow mine. So we became friends because of this Lent bicycle. And at the end of it, he said, would I come to Woods Hole to help him teach the embryology course the next year? So I leapt at the chance because I could work on sea urchin eggs and see whether they might be a good thing to work on. The first year was a complete bust, actually. It turned out that although the embryology class was very well equipped for looking at the cells, it wasn't very well equipped for running gels and doing protein. I mean, they didn't even have a -80 freezer, for example. Certainly no liquid nitrogen. I mean, I thought I would never go back, except that I missed it terribly when I didn't go back for the following year. But I did learn about getting sea urchin eggs. You give them a 12-volt alternating current electric shock. And if it's a female, they shed their eggs. And if it's a male, they shed their sperm. And if you mix them together, then they start synthesizing. There's a big increase in the rate of protein synthesis. This is my very own data. Borsuk was absolutely right. So that was right. But, we began to sort of wonder, why does it turn on? Then went back. I mean, of course, this is actually much more interesting than the biochemical approach, and I did learn to look at them down the microscope. They divide, and they divide incredibly synchronously without you having to do anything fancy. Just mix them together, and they divide synchronously. And the second summer I went, I did some collaboration with Eric and Joan, who were looking at clam eggs, or really clam oocytes, which are shown here, and they don't show much of an increase in the rate of protein synthesis, but they start making new proteins that they weren't making before. So we began to wonder, why do you need to make new proteins? And I should say one thing, which is in the sea urchin case, if you blocked that new protein synthesis, they couldn't divide. So clear implication is that one or more of the newly synthesized proteins are necessary to catalyze cell division. And nobody had really sort of thought properly about that before. And we wondered, what do proteins A, B, and C actually do? Why do you only start making them after fertilization? But we just sort of wondered about it. And it's pretty amazing. The other thing you need to understand, so here is a clam oocyte before and about 20 minutes after fertilization. It's a little bit hard to read. They're both taken with polarizing light microscopy, which sort of reveals the structure, especially of the unfertilized egg. But that structure with the funny bright things is actually the first meiotic spindle, and the bright things are the chromosomes which are being segregated there. And that transition takes place without any new protein synthesis, RNA synthesis, or anything. It entirely depends on modifications to the pre-existing proteins. I mean, you know, the nuclear envelope obviously breaks down completely, and the microtubules are totally reorganized. It's really amazing. And that same summer, there was a wonderful seminar from this guy here, John Gerhardt, about frog oocyte maturation. And if I add some progesterone to these frog oocytes, you'll see that what happens is this white spot appears, and then they do the first meiotic division, and that's a little convulsion and turning over is actually expulsion of the first polar body, throwing away half the DNA. And John explained how Mazui, a few years before, had discovered that what was going on is that progesterone, very slowly, it takes several hours to complete this transition. It's actually a sort of G2 to M, right? Sort of, you can consider it entering mitosis, but it's actually entering meiosis. But if you take an egg which arrests at second meiotic metaphase, and you suck out 50 nanoliters of cytoplasm and inject it into a fresh oocyte, the fresh oocyte now very rapidly matures. It's sort of autocatalytic, very mysterious thing, where this thing that Mazui called MPF, maturation promoting factor, was clearly catalyzing a cell cycle transition. And the key findings were, which Gerhardt described. He was a brilliant biochemist. It was very unstable. It was destroyed by heating and proteases, so it seemed to be a protein. What was it? Well, you couldn't purify it because it was so unstable. You know, the idea that an enzyme to catalyze such a complicated thing as entry into mitosis struck me as really remarkable. And I thought, what a delicious problem. I wish I could work on that. But of course, I was working on this control of protein synthesis problem, so I couldn't work on that. And then over the years, it turned out that this MPF wasn't just in frogs. It was also found first in starfish and then human cells and in yeast. So the pressure on understanding what MPF was sort of over the years gradually got more and more people got to know about it. And then I did an experiment, a very simple experiment, which was simply to fertilize the sea urchin eggs and look at what proteins were made by simple one-dimensional SDS gel electrophoresis. And here is not the first experiment, which I showed yesterday, but the second experiment, where I think you can clearly see, I'm afraid this pointer isn't really so wimpy, you know, this is really a pathetic pointer. Can you? Even I can barely see that, but you see this band here. It's the strongest band early on. It gets stronger and stronger, and then it goes away, and then it comes back again, and then it goes away. And it kept on doing that as long as the cells were dividing, and you could show that if you inhibited DNA synthesis, the cells didn't divide, and the protein didn't go away. So, and indeed, I mean, what it turned out, that this protein is totally stable in interphase and unbelievably unstable at the metaphase to anaphase transition. It's a little bit easier to see here this is clams, and the amazing thing is that Joan and Eric hadn't spotted this. They'd been studying these proteins for 4 or 5 years, and they didn't know that they went away because they never did this really simple experiment of just taking time points. In order to see the dynamics, you have to take time points. And I was always used to taking time points because I was interested in rates, you see. And so I was just sort of an incredibly lucky person who first applied a one-dimensional SDS gel to sea urchin eggs in those 10 years. The people who've done it before didn't have that analytical tool, very simple and powerful analytical tool. Now, obviously, I began to suspect that this had something to do with MPF, but I didn't dare to hope that it actually was MPF. It seemed more likely it was an MPF interconverting enzyme. And one of the things we worried about was how much of these proteins, I mean, these are radioactive bands on gels. I mean, they're tiny, tiny amounts. These are tracer amounts. So, you know, and also, you'll notice that whereas these proteins accumulate linearly, an entry into mitosis is a kind of all or none thing at a particular threshold. So that sort of didn't make sense. But anyway, we persisted. And the trick was to clone and sequence and make antibodies against these things so that we could find out, so we could purify it and find out what it really did. And that took ages. It was very difficult, and I could give a whole seminar about how we did that. Meanwhile, I have a big problem because here I was supposedly an expert on the translational control of protein synthesis, suddenly landing in a field about which I knew absolutely nothing. So what do you do when you find yourself in a field which you're totally ignorant? Well, the answer is you talk to people, you go to seminars, you read the literature, and it turned out that not very much was understood about the cell cycle in 1982, which was lucky for me. So, you know, there are these G1S, G2M business, replication and segregation. So the main thing is getting the two DNA strands into separate. So, you know, this is prepare the tape for copy it, prepare for insert, and put the exact copies, how to make a new cell into the two daughter cells. And interestingly, there are these sort of very interesting, these three, how do you build a switch, an irreversible switch, which have the curious property of the decisions are taken with great sensitivity. I mean, for example, getting out of mitosis, you have to be absolutely sure that all the chromosomes are properly lined up. So how the hell do you ensure that chromosomes are all properly lined up before you exit mitosis? So these and many other interesting problems have all now been solved, but they were all totally unknown at the time because we just had no way of getting in at them. And the only progress that had made up to that point was by the yeast geneticists, and here's Lee Hartwell, who really focused his attention on CDC28, cell division cycle mutant 28, because that seemed to be a regulatory gene, because it controlled very different processes. I mean, spindle pole body duplication and DNA replication and the formation of a bud. And Paul Nurse read Lee's papers and started working on a different kind of yeast, and he focused on CDC2 because it was a gene that could be knocked out, in which case the cells didn't divide, they just got longer and longer and longer. But it also mutated to an allele which accelerated, so he argued that must mean it was a regulatory thing. And then the amazing thing was that they seemed to be the same thing. And progress really came from frogs. I mean, one of the things about this field was that those early pioneers, I knew all of them pretty well. You know, I worked on sea urchins and clams. Paul worked on fission yeast. Lee worked on budding yeast. Other people worked on fruit flies. What else was there? I mean, some people worked on mold, and we all sort of saw the problem from a slightly different angle, and there was no real competition between us because none of us had a clue what was going on. But we loved to get together to chat and try to figure out what the hell was actually going on. And the real breakthrough came from making a cell-free system, rather like the reticulocyte lysate, from frog eggs. If you spin the frog eggs, you get this beautiful yellow cytoplasm, which will, if you add nuclei to it, know exactly what to do. It's egg cytoplasm, so it assembles a second meiotic spindle. And using that as their assay, Fred and Jim managed to purify MPF, and there's the protein gel is on the left. On the right-hand side is a kinase assay. The protein kinase had two subunits, and you didn't have to be a genius to suspect that the small subunit is CDC2, protein kinase by sequence, although if you purified it, had no activity. The larger band was cyclin, and we proved that, and so that was the answer. And the complex has protein kinase activity, so it's really easy. It's another protein kinase, and without cyclin on the right, the CDC2 has absolutely no activity. So it's really easy. You accumulate cyclin, CDC2 turns on, you phosphorylate lots of proteins, and you enter mitosis. I mean, obviously that begs lots of questions. How does it do that? But a problem that sort of then we sort of skip forward from about 1992 to 2005 or something like that, a very long period where I got completely lost and almost lost my job because things were going so badly. I got a wonderful postdoc in the lab who we started investigating this thing. If it's true that you enter mitosis by sticking lots of phosphates on, it follows that you must exit mitosis by taking all those phosphates off. And even there, early on, there was a problem because the geneticists all said that protein phosphatase 1 was the important enzyme. The biochemists all said that protein phosphatase 2. Turns out, of course, that both were correct, and it just depends on what kind of assay you used. But as a biochemist, one of the first things you learn in biochemistry is about futile cycles, and I sort of love the idea of a futile cycle. Basically, if you have a protein kinase phosphorylating substrates and a phosphatase taking them off, you can't go anywhere. That won't generate a switch. What you've got to do is have either one or the other and not both at once. I mean, it's very simple. If you want to fill your wash basin with water, not only do you have to turn on the tap, but you must also put in the plug. And likewise, when you want to empty it, you mustn't just turn off the tap. You have to open a plug. So if you think of the tap as the kinase and the plug as the phosphatase, that's absolutely right. So what was going on? Well, I had this wonderful postdoc, and the final lesson really is that by this time, you know, I'm a Nobel Laureate and very famous and stuff, but Satoru and I had this wonderful level relationship together. That, you know, I could suggest he do an experiment, and if he didn't like it, he could say, no, Tim, that's a silly idea. And he wasn't in the least bit intimidated. I wasn't intimidated by him either, because he was an absolute genius in the lab, a wonderful, wonderful guy, actually. And we had such fun pursuing this story. And what we found in the end, we discovered, I mean, this is a summary of about 5 years' work. What we discovered was this little thing here called ENSA, which is an inhibitor of PP2. And these feedback loops, I mean, actually there are 2 positive feedback loops in here. I should mention one other Hungarian thing. So in analyzing this, we had a wonderful collaborator called Béla Novak, who actually came from Budapest, and sadly his heart stopped beating about a month ago, and he is no longer with us. And I miss him terribly because he understood all this stuff really, really well and could put, you know, differential equations and analyze it in detail. And the predictions, finally, he and Satoru collaborated, and they could really make beautiful models of this that very accurately represented how the system works. So you look at that, it's really not, you know, it's very hard to make sense, even in such a simple circuit, what's going on, and they could do it. So, you know, there's a huge amount of stuff that I've left out, but I hope I've sort of given you an impression of how it goes. I mean, you know, you get stuck, terrible things happen, and you just have to sort of keep going, but you never know where you're going. So good luck, everybody. Thanks.
Thank you, Tim, for the wonderful talk. Now we have a few minutes for the questions, so the floor is open for the 1 or maybe 2 questions.
Does anybody have a question? You must all be exhausted by now, and I was so depressed by David's talk, too. You know, I worked in a cancer institute for ages, and we never ever discovered cancer. I mean, I once saw a tongue cancer. It was really horrible. It had just recently been cut out. The poor fellow had lost his tongue, you know. It was, oh, dear, 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 dear. Comment? Yes.
Yes.
Yeah.
Thank you, Professor. My question is about what do you think is the most important trait and habit for a young researcher, and how can we cultivate it? Thank you so much.
So, yeah. What does it take? What does it take to be a successful scientist? And I don't really know. I mean, I think I read a book recently, and he said he'd often asked this question. He was a physicist. He said that curiosity, number 1, and determination, number 2. I would say a certain obsessive quality. I mean, you know, that when you're on the track of something, to be absolutely resolute about, you know, trying to get to the bottom of it. And don't forget, the scientists are very competitive people. You know, we don't like to be beaten. We like to be first. No use being second. So there's that aspect to things as well. So you just have to sort of keep going. But I think above all, you just want to know once you've got your teeth into a good problem, you really want to know what the answer is. It's sort of like a long-standing detective story, you know? And you will get stuck. You always get stuck. You will make mistakes. And you have to want to get to the bottom of things enough so that you will keep going. So I would say just keep going. I'm very often asked that question, actually, you know, what do you do when you get stuck? And the answer is, of course, if you're pretty sure that what you're doing always gives the same result, which it should do. I mean, sometimes it's technical, you know, but by the way, if it is technical, you know, you're trying to do something that's difficult, everybody loves to teach people things. So if you're having difficulty with the technique, go to the lab where they do it and just spend a week there to see how they do it. Because usually you learn sort of little tricks that they don't actually write in the materials and methods. And it's a good way of meeting friends. And I can't emphasize how important it is to get out and meet people. I used to have funny arguments with people in Woods Hole. That a lot of supervisors said, you know, I'm not going to let my students go for 2 or 3 months, because they've got a project to work on. Well, whenever I sent a student to Woods Hole, they came back so much better as a result of having worked with their peers from all over the world and seen how easy it was and how quick it was to learn a new technique if you simply knew what you were being taught by somebody who knew how to do it. Easily made up for the month or 2 that they'd spent away. Very important, that. All right.
Any more questions?
Should we go on? I think we should go on.
Yeah. Then before that, it's on behalf of Semmelweis University. Professor Hunt, thank you so much for your contribution. It's my pleasure to give you a certificate of appreciation for the seminar lecture series. Thank you so much for your time.
Very good.